Hello everyone, welcome to VetGuru. This is your tutor Dr. Dixit Kumar. So in the last lectures we discussed about what is parasitology, what are the different type of parasite, what are the different type of host, how they relate with each other, or to control them, what are the different type of drugs that are used, what are their mechanism of actions. So at the last I told ki we have that much of the drugs, but still we encounter most of the problems of parasitic infection in the field. This is because these parasites acquire resistance against different drugs, and our immunity can't cope up with that, okay, or the animal immunity can't cope up with that, okay. So in this lecture we are going to discuss about that portion: parasitic immunity, what is anti-helminthic resistance, how we can diagnose different type of parasitic infections, and instead of drugs what are the other biological methods to control the parasites, okay. So if we talk about parasitic immunity, so as you know, the basic concept is, suppose if any parasitic infection occurs in the body, there will be release of IgE. Okay, so these IgE will bind to parasitic surface, they recruit immune cells, and then they causes destruction of parasites. So this is the usual cycle that will occurs in the body defense system. A parasite will come, IgE will be released in response after antigen recognition, then they will recruit different type of immune cells, and then will cause destruction of. Instead of this immunity, there are other specialized immunity that are maintained in parasitic infections. So what are those different type of immunities? First one is sterilizing immunity. Sterilizing immunity is a specialized type of immunity in which the immunity that persists even in absence of parasite. Suppose coccidiosis infection occurred in the body, bird or any of the calf you take, parasite came, it infect the body, but suppose the parasite reaction is over, parasite is shedded from the feces, still now also immunity is maintained in animal body. So such type of immunity is known as sterilizing immunity. Even after the parasite is removed. Okay, so such type of immunity, or you can tell it as long lasting immunity. So this immunity is known as sterilizing immunity. Reverse to it is the pre-immunity. Pre-immunity means this immunity is maintained only in presence of parasite. Until the parasite is present, only then immunity is maintained. Means parasite will not be eliminated, the parasite population will be controlled, because due to the reaction of parasite, IgE or other cells have released in the body, so they are maintaining the immunity. So immunity is maintained up to a level only. If parasite is removed, again the immunity will slow down. Okay, so such type of immunity is known as pre-immunity. This type of immunity is basically seen in babesiosis and filariosis diseases. Then comes the another part of this pre-immunity is concomitant immunity. Concomitant immunity means in this condition the host will be resistant to new infection while still harboring the original pathogen. Suppose a parasite is multiplying in the body and it formed its stages. Let's suppose fasciola is there. Okay, so until fasciola is there, no any infection can occur in the body. So such type of immunity is known as. Okay, so original pathogen is present, new infection is not occurring. Okay, so such type of immunity is known as concomitant immunity. Best example is resistance against new larva but not against existing adult. Means the new larva can't infect the body because already adults are present in the body. Then comes the cross immunity, as the name suggests, means immunity against one parasite will confirm immunity against another. जैसे कि fasciola hepatica immunity, fasciola, so that time it will not be affected by amphistome, so that is known as the cross immunity. Same, suppose a host will acquire resistance against Taenia ovis antigen, so that time he will also be protected against Taenia solium. Okay, so these are different type of immunity that are seen in the body against parasite. But these parasites are also very clever, they will also develop some resistance or some different type of resistance. Okay, so what are those resistance in response to these different type of immunities? One comes the multiple resistance. Multiple resistance means the parasite develop resistance against two unrelated drugs with different mode of action. Suppose if we are talking about the any of the parasite to take, like we are taking fasciola again. Okay, so suppose we thought, okay, due to continuous use of benzimidazole, fasciola is resistant to benzimidazole. After reaction, you are seeing that, okay, this parasite also acquired resistance against ivermectin. So both benzimidazole and ivermectin are different class of drug, different mode of reaction. In that manner that it will acquire resistance against both the drugs. So such type of resistance is known as multiple resistance. Then comes the side resistance. So side resistance is, suppose bacteria develop resistance against one drug. So that resistance, that mechanism will provide resistance to another drug also. Luckily, so that type of this is a type of multiple resistance only. It is due to one drug. Means here parasite is resistant to ivermectins only. It changed its molecule in that manner that it acquired resistance against benzimidazole also. Okay, so that is known as side resistance. Okay, then comes the important phenomenon, that is reversion. Reversion means these parasites will lose resistance when the drug use is stopped. Like suppose if you talk about benzimidazole, we are continuously giving benzimidazole to an animal. So parasite that Haemococcus contortus develop resistance against benzimidazole. But suppose if we remove the use of benzimidazole, then parasite will also lose resistance to benzimidazole. So suppose if after some time you are using benzimidazole again, that time benzimidazole may be effective. So such type of phenomenon is known as reversion. Okay. Then how we can measure this resistance? Bacteria, parasite acquired resistance or not. So how we can measure it? So there are two type of method. One is in vivo method and another is in vitro method. So in vivo method, best example is fecal egg count reduction test. So as the name suggests, fecal egg count reduction means in the presence of drug, how much count of fecal egg is reduced. Okay, so suppose after administering benzimidazole, if the egg count of feces is reduced, it means the drug is working very well and parasite does not acquire resistance. Okay, so this method is known as FECRT, fecal egg count reduction test. Many times question is asked in the exam. Okay, so basic mechanism you remember. You have to compare the egg count before and after treatment. Before treatment, before giving the benzimidazole, how much eggs were releasing in the feces, and after treatment, how much eggs are releasing in the feces. So that will help to compare. Okay, then comes in vitro test like egg hatch assay, means you can analyze the hatching of egg in presence of drug. Like suppose in presence of drug also eggs are hatching, means drug is not working well. Okay, same larval development test. So suppose in the presence of drug larva died, means the drug is very well, the larva didn't develop any resistance. But if it is growing in presence of drug also, means your drug is waste. Then same larval motility test, we can check the motility of larva, or otherwise we can use other tests like molecular assay techniques like PCR or ELISA. Okay, so these are different ways to measure the anti-helminthic resistance. Now the main point comes, why these parasites acquire anti-helminthic resistance. Okay, so one major thing that is frequent treatment. Means suppose you are continuously using overdosing the drug, so the strains with whatever the parasite will there, they will mutate themselves and they will resist themselves. Another is underdosing. Underdosing is also harmful because suppose if you are giving benzimidazole, you thought okay infection is controlled, but some population of parasite was hidden and it remained in the what you can tell, it remained in the body. So that time resistant ones or the survival ones, they will proliferate, and in their progeny their characters will pass, so they will develop resistance against those benzimidazoles or any other drugs. So both underdosing and overdosing are harmful, or the factors that may contribute to anti-helminthic resistance. Okay, like I told you, underdosing what you learned. Okay, parasite, whatever the resistant parasite are there, they will proliferate. So such type of phenomenon is seen in parasite, that is known as self-cure phenomenon. It is similar to that. What will happen, suppose a sheep population or an animal population is continuously getting affected by a parasite. Let's take example of Haemococcus contortus. Okay, so what will happen due to avoid overloading, whatever the adult worms will be there, they will be expelled from the gastrointestinal tract to make space for the new larva. Okay, so that condition is known as self-cure phenomenon. Okay, but this condition occurs only after repeated exposure. For primary infection, it is not applicable. Only after repeated infection, it is seen, and it develops due to type 1 hypersensitivity and triggered by L3 larva. L3 larva will molt into L4. Okay, so this is the how self-cure mechanism works. Many times question is asked in the examination, what is self-cure phenomenon, by which type of larva it is triggered. Okay, so you remember these points. Now whatever these parasitic infections are there, like we learned, okay, parasite is there, it is causing infection, it may register it, if suppose if it's developing resistance, means it is acquiring infection. So how we can diagnose those parasitic infections? Okay, so first one comes the rapid or the field friendly test. So lateral flow assay means rapid immunoassay. You can detect the parasite by a labeled antigen. Suppose if you are suspected that okay, my animal may be affected with hemoncus, so either you can, so obviously in blood antibody must be formed against hemoncus, so you can take a hemoncus antigen and catalyze the immunoassay, so antigen antibody will bind and the colored reaction will confirm that the infection is present. Okay, same latex agglutination test, it is also similar to immunoassay, means we can use latex bead coated with antigen or antibody. So either if you want to diagnose antigen, then you can use the latex bead that are coated with antibody, or suppose if you want to isolate antibody, then you can use antigens. Okay, so whatever the visible clumping is there, the clumping will occur if infection is present, clumping will occur between antigen and antibody, visible reactions will produce, so that will confirm that okay, parasitic infection is present. Instead of that, we can go for the fecal examination also. So there are two methods of fecal examination. Either you can go for flotation or you can go for sedimentation. Like suppose you collected feces of an animal. Okay, so one method is you use whatever the solution in which you are going to mix the feces, that should be of higher specific gravity. So suppose if the solution is of higher gravity, the solution will settle down and the feces or the eggs will float upside. So that technique is known as flotation technique, means we are allowing the feces or egg to float on the surface. So there are different techniques for that. Either you can use Wills technique, or you can use lens technique, or basically you can use the McMaster technique, that is a quantitative method for egg count. Reverse to that, suppose if you assume that okay, maybe the eggs may be of higher specific gravity, so that time you use a solution of lighter specific gravity, so in that the heavier eggs will settle at the bottom and solution will go upside. So basically this is used for the heavier trematodes eggs like Fasciola hepatica, they produce giant eggs, so that time we can use sedimentation technique. Instead of that, there are other blood and serological tests like ELISA is used to detect parasitic antigen or antibody, or like a NOT test is used for diarrhea immitis. Instead of that, there are other tests like if you talk about the Trypanosoma evansii infection, so card agglutination test is used. So that is also a type of agglutination test. One antigen, one antibody will be there. They will bind and produce agglutination. Then still Barmedine test for Trypanosoma evansii infection. This table you keep in mind because most of the times direct question are asked. What is the diagnostic test? Then here they will give you the parasite. Then you have to do the matching. Okay. Then PAT check test, that is used for Diarrhea immitis infection. Then NOT test, that is also used against Diarrhea immitis infection in dog. Test is used for Toxoplasma gondii. Toxoplasma gondii is the favorite question of an examiner from protozoology. In matching, directly they will put torch test, Toxoplasma gondii. Okay. Then Cossoni skin test for Echinococcus hydrotis, Echinococcus infection, hydatidosis. We have to isolate the antigen and then the intradermal reactions are produced. Then capillary tube test for anaplasmosis. Bartlett test for trichomoniasis. Beckman test for trichinosis or trichinellosis. Then Montegro test for leishmaniasis. Or you can use Cercarial hural reaction for schistosomiasis, because schistosomes they produce Cercarial dermatitis. So in that case, you can use those reactions or those tests. Instead of that, you can use some molecular tests like PCR that detect parasitic DNA directly, and they are considered more sensitive and specific. Okay, you can design specific primers, and then DNA will bind and the reactions will produce. Okay. So here now you know, okay, there are different type of parasitic reactions are occur, and we can diagnose the parasitic reactions. Okay, so like I told earlier only, there are different type of drugs, but instead of drugs, there are other methods to control the parasite. So that is known as the biological control of parasite. Means here we are, instead of drug, we are using some biological species for the control of parasite. Okay, so first one is nematophagous fungi. Nematophagous fungi means the fungi that is phagocytic on the nematodes. Okay, they are directly eating the nematodes. Okay, best example is Duodenothonia flagrans. Okay, so that is effective against Hemoncus contortus in ruminants. Arthropotaurus polygospora, that is also used against nematodes. Then Purpurocilium species that are also used against nematodes. One of the question is asked many times in the exam. In 22 also it was asked Pilobolus fungi. So Pilobolus fungi is used against the Diclococcus viviparus species. That lungworm is there. So they will directly ask you the following fungi is used for the parasites. So just remember two or three names of fungi. Okay. Then there are some insect-eating fungi that are called as entomopathogenic fungi that are pathogenic on insect. Okay, like Beauveria bassiana, Metarhizium ensicolia, and Lacinium lecani. So these are entomopathogenic fungi that are phagocytic on insects or that are pathogenic for insect. Then there are bacteria also like Bacillus thuringiensis that will produce toxin proteins against insect larvae and some nematodes. Okay, so these are the different ways how we can control the parasite. Okay, so in this first unit of general parasitology, we discussed about what are the parasite, what is the historical origin of this parasitology. Okay, then after that we discuss what are the different type of parasite, how their life cycle proceed, how their body systems develop. Since parasite is there, definitely there will be a host. So what are the different type of host, what are the associations between them? As a veterinarian, how we can control the parasites? So what are the different type of anti-helminthic drugs, what are the drug of choice for specific disease? Okay, instead of drugs, there is possibility that there may be parasitic infection. This is because these drugs, these parasite acquire anti-helminthic resistance. So what are the different type of anti-helminthic resistance, how body pop up with that resistance? So what are the different type of immunities, how we can diagnose these parasitic infection, and how we can biological control parasitic infection. Okay, so this was all about today's lecture and this unit. Thank you, have a good day.